Yeah, okay. Thanks, Chris, for the nice introduction. Hello, everybody. Nice to see you or hear you. And I'm very impressed by you guys that you do this evening course after all your daily business. So I'm recognize I'm running out of energy at this time of the day, but we will do our best here. So today's topic, microbiology of the biogas process. And no, it's not moving, Chris. No, it's moving. Great. A short outline, what I will cover in this lecture is biogas, what it is and where it is produced. General features about anaerobic digestion processes. That it's a way for a sustainable way for energy production and using renewable resources. And I put a question mark here because this is not always the case. And we will see that when we look at different countries and how they perform biogas processes. And we will take a look on the microbiology of the biogas process. And then I want to talk about syntrophy, which is a special microbial lifestyle and which is quite important for the biogas process. And there it might be get a bit more complicated, but I hope that I can make myself clear and make it understandable how it works. What are these special features of these microorganisms? Biogas is around mankind since its rise, and you see here this. What I want to show you is like a peatland or a swamp where you can see flames, and because the biogas itself, it's very easy and flammable. And in former times, people didn't recognize where it come from, so it actually scares them because they were thinking these might be ghosts. And they also were misleading travelers in the night because they were thinking there were maybe fire houses with the fire. So they left their track and actually moved just deeper into the peatlands and maybe even came to dead. Later on, they recognized that this originated from the decomposition of organic matter. And it was actually Alessandro Volta who discovered 1776 that this is a methane, what is inflammable. And he did it by here, as you can see on the picture. He collected tiny bubbles from muddy waters, and then he transferred it, he turned it, and then he inflamed it. And he called it inflammable air native to marshes. Methane itself is colorless and it is odorless. So it doesn't smell at all. And many people doesn't know that it doesn't have a smell. Biogas is produced by microorganisms in environments where you have very little or no oxygen. So in anaerobic or anoxic environments. And you also have to have no other alternative electron acceptor. So no sulfate, no nitrate. If this is not there, you have methane production. And biogas consists mainly of methane and carbon dioxide. And you can find it in sediments and wetlands, in ruminants, in particular cattle. They are a high producer of methane, as you know. Termites, there's a big source of methane production. Of course, rice fields, they release a lot of methane, and also a big part of the human population, actually. And this is something I would like you to be aware of that, but this is an increasing situation actually, because the methane production in humans, it's often connected to increasing obesity. And we know that obesity is something we have problems in certain regions on this earth, in certain populations. So these all environments producing biogas, producing methane. Mankind started using biogas a long time ago. So there's evidence that already 1,000 years before Christus, they used biogas for heating bath water. And also Marco Polo described on his travels that there were covered sewage tanks in China already in the 13th century. And China itself could track it down probably far back as 1300 before Christmas, that they used already constructed crude digesters, which what they're called Vasi. Nowadays, we use, at least in our hemisphere, quite controlled anaerobic digestion processes. Here we control the breakdown of organic material to biogas and use this as an energy source. And you can start this process by naturally occurring microorganisms, which you can find in manure and slurries. These biogas systems, they can look actually quite simple, and you can find them all over the world. And this is an example from a project in Kenya, where they started to establish a lot of these house-scale biogas systems. They are quite simple built. So you have a kind of an inlet, and then you have a kind of a, like an arc built. On the top, it collects the biogas. And here you can take out the residues which are left and use it as a fertilizer and put it on the field. And the gas you can use for cooking or also generate electricity. In particular, in this country, it helped a lot to improve sanitation, to avoid the spreading of diseases, to get energy at low costs and make farming more easier. You have other simple systems. You can find large, small, mini, micro-scale solutions. Here, example from Rwanda, how they look. Here you have reactors in Thailand. Here you see one in Bangladesh, where you see the connection to the house, where they collect the gas for cooking and heating if they need it. And here's one in China, and China has most of these domestic biogas reactors, more than 30 million. And they used it quite early. They established those systems very early. Most of them they start building in the 50s and 60s to provide the population with energy. Here's an example from also from Kenya. That was, I found it. It was a report from an engineer who traveled to China and wanted to build up quite simple systems what they can use. So, and this is just a container which produces the methane from a slurry. Then it's guided, the methane is guided through two systems for purification and for enrichment of the methane. And then at the end, he wanted here to use for gas for the households. But when he came there, it's not that they have a Bauhaus around the corner. So this was actually the local Bauhaus he found. So there's not much material he found he could build on. He could build with this small construction here. And at the end, it is as simple as it looked like and what the people can use and partly have to use. It's simply a big a can, and you collect the biogas and connect it to very simple heating systems. Here was an example from students. They wanted to show when you have slurries in a simple water canister that it inflates over time the tire here to demonstrate that gas is evolving. So these are our systems, how it looks for us in the more or higher developed countries. This is the example here for Newt-Berger. That's a large-scale biogas plant. Here you have a farm-scale biogas plant on Skaraborg. Here you have farm-scale biogas reactors. In Germany, you find many of those. And this was once a student project who built a house-scale biogas plant, and he even converted that and upgraded to fuel to fill the car with that. So biogas itself is a versatile and renewable energy source. And we can use a different substrate, what makes it renewable. We can use agricultural waste. We can use food waste and industrial waste from food industry, energy crops, and sewage sludge. These all is digested in the biogas process and produces biogas. What is left from this digestion process can be brought back to the fields and can be used as a fertilizer. The biogas you can convert to electricity. You can use it for heat, or you can upgrade it to biomethane. That means it has a higher methane content at the end, what you need to drive buses. Here you see the nice green bus from Uppsala. They're all around. Or you can fed it into a gas grid and use it for cooking. And that makes the versatility of the process, that you have different forms of energy you can use. I want to tell you a few numbers from the biogas production in Sweden and what it means for us here in Sweden to use the technology. We have around 280 facilities. Maybe there have become a few more, but I actually don't think so, because these numbers are from 2015. And most of them are wastewater treatment plants where we treat the sewage. We have a few landfills left. We have around 40 farm scale plants. We have co-digestion plants, which using household waste and mixed with industrial wastes or restaurant waste. And we have a few other industrial plants connected to the industry and a few gasification plants. The feedstock we are using is mainly sewage sludge. We're using manure, industrial food waste, a very, very small proportion of crops, as this is not allowed to use in Sweden. And they only can use it in the farm scale plants to balance a little bit with the manure, that the nitrogen carbon balance fits. A few others, whatever they consume, it didn't tell me. And then we have around 4% of organic household waste, what we use. Around 81% of the digestate produced is used as a fertilizer, so it's brought back to the fields. And what we do with that, it's mainly vehicle fuel. Here you see this large filling stations in the center of Uppsala. It's around Kungsengen. Here you see how the buses charge their gas tanks or load their gas tanks. You use 20% for heat, a very small proportion for electricity. A small proportion is used by industry for different chemical processes. We always lose a little bit by just burning it. And then here's the rest of 2% what disappear on different ways in different ways. When you look what that means for us of the whole process, it's a recirculation of energy and nutrient from urban and agricultural organic waste streams. That means that the waste produced from the cities, either by food industry or by household, and the waste streams produced in the agricultural sites goes into the biogas plants. These produce the biogas, and the digestate, what is left from the process, goes back to the fields and is used as a fertilizer. They produce on the agricultural sites food again, which goes to the city, and so on and so on. It's a recirculation. And it has several benefits. It has a waste management, so we treat our waste. It reduces the greenhouse gas, and you have a feedstock at the same time for the biogas plants. When you use the biogas, you can replace the fossil fuel in all energy sectors for power, heat, and vehicle fuel. You recycle the nutrients back to the soils, and you can replace mineral fertilizer by that. So when you look on that, you would think this is a sustainable way. Now we want to look in another country. We look at the biogas production in Germany. How is it here? They have nowadays more than 10,000 biogas plants. Most of them are farm scale. Just very few upgrade to biomethane, and they produce a lot of electricity from that. All the green dots here it's a biogas. It's a farm scale biogas in the mean here, spread over the country. What they use is different from that what we use. They use half of it energy crops. And the other half is agricultural residues, mainly manure. And very little is, for example, household waste or industrial waste. They produce mainly electricity from that, very little heat and very little transport fuel. And when you look at this, you will think it is renewable. Yes, you use renewable resources, but is it sustainable? And when we go back to that circle, what I showed you before, you see that actually there's no recirculation of energy and nutrients because this circles only exist only here. So you breed, you grow energy crops, put it in the biogas plants, produce biogas from that, and the residues go back as well, but they don't produce food in that sense anymore because they use it only for the energy and they almost not treat the waste. This had actually consequences on farming. Before, these farmers were food producers. So they were cereals and vegetable farmers, and then they turned into energy farmers. Instead, they started using maize and did not produce food anymore. And that looks like this. That looks as ugly as it looks here on the picture, actually. These landscapes are incredible dead. And it had consequences for nature and also the public health. We had, for example, a striking drop in the overall insect biomass. Here is a picture. They compared 89 to 2013, and they saw that the insects they collected were incredibly less compared to 1989. They have no crop rotation anymore. They have a use of insecticides and herbicides and pesticides. They actually use more land than they would use for vegetables. This has something to do with regulations. You can cultivate the maize in humid areas where traditional crops cannot grow, and they over-fertilize the soil. As maize can take a lot of more fertilizer than other plants. That had an effect of pollination of croplands. 70% of the croplands needs pollination by insects. It also had an impact on the bird population by almost, this population dropped by almost 80%. And they also started to accumulate high nitrate in the groundwater and in the drinking water. And the next thing is that the maize cultivation itself is not a climate neutral process because it releases more carbon dioxide from the soil, what has something to do with how they treat the soil. It reduces the seed diversity in soil, and also actually tourism started to be affected because these landscapes were dead and there were no noises anymore and no one traveled there any longer. And that is what I want to show you by this comparison, that the production and use of renewable energies does not automatically mean that it's a sustainable process. In that case, you lose biodiversity, what have an effect on other communities. Let's come to the microbiology of the biogas process. The biogas process requires a combined activity of many diverse microorganisms. There are various microbial degradation pathways which needs to be harmonized, that we can ensure an efficient metabolic flux between the pathways, between the microorganisms. Here's the anaerobic food chain in an engineered AD process, how it looks like. We have 4 steps altogether. They are called hydrolysis, fermentation, anaerobic oxidation, and methanogenesis. The hydrolysis happens at first. Here you degrade biopolymers like polysaccharides and proteins to smaller compounds, to oligomers and monomers. This is done by fermenting bacteria, but also by facultative anaerobes. And these smaller compounds are further degraded to so-called intermediates. They are short-chain fatty acids, alcohols, lactate, for example. This is partly done by the same bacteria as here, but also by other bacteria, which cannot the first step, but they can use the smaller compounds. And you have these intermediates, they are further degraded to carbon dioxide, hydrogen, and formate and acetate by so-called syntrophic bacteria. You have also a facultative syntrophy, which makes that you actually circumvent these intermediates. They produce directly acetate and carbon dioxide and hydrogen from the smaller compounds. And here is the methanogenic step. The acetate is converted by acid-clastic methanogens to methane, and the CO2 and the hydrogen, and also the formate is converted by so-called hydrogen-trophic methanogens. These methanogens, they are archaea, they are not bacteria. And here you have also syntrophic bacteria, which can convert acetate directly to CO2 and hydrogen, and also the other way around by acetogens. So you see there are a lot of different microorganisms, a lot of different metabolism in this food chain. We look a little bit more detailed on the steps. What happens there? The first step here was the hydrolysis. This is performed by many specialists, and these specialists, they excrete a lot of these extracellular enzymes into the environment. Partly they keep it fixed on their cell surface. These are enzymes like amylases or peptidases. They can cleave these large complex organic molecules. This is not very easy and it takes time. It is a slow process. And from the large molecules, you make smaller ones, and then even smaller to oligomers. And these oligomers are further digested to monomers or dimers. And then it goes faster and faster because the molecules are getting smaller, they can get picked up by the cells, and they're not as complex anymore. These complex organic materials is, for example, quite abundant in plant residues, for example, cellulose. From that process, you get sugars, amino acids, fatty acids. They go into the next step. It's the fermentation step. Here, these sugar and amino acids, they are further converted by fermenting bacteria. Fermentation, you know, probably from yogurt production, cheese production, and also ethanol production. It simply means that you degrade organic compounds in the absence of oxygen. This is what fermentation means. And here you see different bacteria involved. This is a step, you have a lot of variation in the bacterial community. Here, for example, lactobacillus, they produce lactic acid, but also acetic acid. You have bacillus, they produce butanediol, they can produce ethanol, propionic acid, butyric acid. So here is a lot of activities and a very diverse metabolism behind. And from that, sugar and amino acids, they produce these different kind of organic acids, alcohols, lactate, but also hydrogen and carbon dioxide. The next step is the so-called anaerobic oxidation. These steps produce at the end acetate, hydrogen, and carbon dioxide. And these steps require syntrophic corporations. I will come to that on next slides. The last step is the so-called methanogenesis, what produces the energy-rich methane. And here, the methanogens, the acetylclastic ones, the hydrogenotropic ones, they convert the hydrogen and carbon dioxide and acetate to methane and carbon dioxide. And this is what at the end should come out of a process. We have these two different groups of archaea. They are quite specialized. They use almost only hydrogen, formate, and acetate, and very few other compounds I have not listed here. They grow extremely slow. They can restrict the speed of the biogas process because they grow so slow, and they are very, very oxygen sensitive. Here's a picture. They have an autofluorescence. You can recognize them in a microscope as this light spots here. So altogether, that this process is a harmonic process. You have many parameters which influence an optimal biogas process. You have, of course, the time. You can have short process, longer process, depending on what you feed into the process. You can have, it can happen that the food is too complex, then you will not get a good process. You can put too much, you can put too less food. You have different temperatures you have to decide on. Will you use mesophilic temperatures around 37 degrees or somewhere between higher temperatures? We have processes that are running above 50 degrees, which is called a thermophilic process, and where you have organisms specialized on this high temperature. They can, for example, not perform at these temperatures. You have a pH, which can affect a lot of microorganisms. A few like a neutral pH, other can take variations in the pH. They can take an acidic pH, and others cannot at all. So you have to have a good balance here. And of course, you can have inhibiting components like ammonia, which can also disturb the process. So that means different microbes react differently and they have different demands. And when you have an inhibition or one of these parameters doesn't fit, you can have a total collapse of the whole process. Sometimes it's wanted that you don't want to have the entire process. That means you want to have, for example, disintermediary products. You want to produce fatty acids or lactate, and this happens simply by high organic load. That means you simply overfed the process. And what happens then is that it promotes fast-growing hydrolytic and fermenting bacteria, so these two steps. But the slow-growing methanogens, which performing this step, they get washed out because they cannot take the speed as this is produced here. That means that the hydrogen content increases and also the acids content increased, and there's no longer methane production. But from that acids, for example, you can use that as a building block for polymers like plastic. So this is an ongoing application. There's going on research all the time, and it has been used to replace fossil resources and replace this by acetate produced, for example, from anaerobic digestion. So summarized, you have a lot of process parameters. You have process types, which are differently. You have different substrate composition, what you put in. You have different operational parameters, like how much do you feed, what is the temperature. You have inhibitory substances. And these process parameters impact on the composition of the microcommunity and the other way around. This this microbial community, the composition, defines the function and the activities you have. And those, of course, impact on the efficiency of the whole process. And you have to get this in balance to get a high productive and stable process at the end. So, syntrophy. What is syntrophy and why is that special lifestyle so important for the anaerobic food chain? We take this picture here again. Here is where syntrophy occurs. And what does syntrophy actually mean? And syntrophy means translated working together or eating together. It means it is a cross-feeding between two partners. Here, an example, two nice partners. And the point with that is that one organism is degrading a substrate and the other is using the products. And another point is this substrate cannot be degraded by one of the organisms alone. They have to work together. If they're not together, this substrate is not degraded at all. That means they have to combine the metabolic activities to degrade a substrate. Then we have also a facultative syntrophy. I just want to mention it to make it complete. I go back here. The facultative syntrophy occurs here. It means that they can degrade a substrate alone, but they have a benefit when they're working together with other microorganisms. It means that they can perform another way of metabolism that gives them more energy. And you also get other products than you got before. So, but we talk about this obligate syntrophy here. So what does syntrophy does? And now it becomes a little bit more complicated, I think, but I hope I can explain. A syntrophy makes unfavorable reactions favorable. Let's have a look here. A favorable reaction happens spontaneously. So like this is a waterfall and the water goes down the waterfall itself. And also a ball, what you might put here, it just simply goes down. This happens spontaneously. And this is expressed by this term. When there is a minus, it means there is energy what you release. Because when that's water what goes down the waterfall, you could do work. Then you have the opposite. If the ball is down here or the water is down, you have to put work into the system to get the water back on that level. So you need energy for that. You don't gain energy from that. And then you have somewhere in the middle, this is the equilibrium where you can, reaction can happen into both directions easily. If you have to put energy in a system, it's a plus. If you gain energy from a system, it's a minus. Here I have an example what syntrophy does. It combines spontaneous reaction, which release energy, which reactions which are not spontaneously, and where you need energy for. If you can release more energy here and you need little energy here, you have still energy left altogether. What can be used by the bacteria? On the next slide, I have an example for acetate oxidation. You have an acetate oxidizing bacterium, which cannot oxidize the acetate. Generally, acetate is oxidized to carbon dioxide and hydrogen, but this doesn't happen at all spontaneously. This is by the plus marked. This reaction cannot proceed itself because you have to push the reactions toward up the waterfall. And then you have methanogens, the archaea. They can take carbon dioxide and hydrogen in producing methane. And this is a process which runs very easily. This releases energy, as indicated by the minus here. And what they do, they combine their activities. The acetate oxidizing bacteria is degrading the acetate to carbon dioxide and hydrogen, and the hydrogen is picked up by the methanogen, where it's used to reduce the carbon dioxide to methane. And what actually happens is that the hydrogen is continuously taken out of this reaction, and that pulls the overall reaction into that direction. What you also can see here that they release altogether very little energy. Here I put the example of a glucose oxidation under aerobic condition. It releases a lot of energy. They release very, very little, and they have even to share this energy. And this is what makes them so slow growing. There is not much energy they can grow for. When it comes to isolation and co-cultivation of these syntrophs, if you want to study them, that's very difficult actually, because they have very strict cultivation requirements. You have to cultivate them without oxygen in a reduced environment. And it's also difficult to find a substrate. So if they need each other, how can you isolate them from each other? That means it is quite difficult to reconstitute this acetate oxidation process in a laboratory condition. Here's an example how you can envision the whole isolation process. You have here lab scale biogas reactors, so small copies of the large biogas plants. Then you take a sludge sample, put it in this anaerobic bottle under anaerobic condition, put a substrate, let's take acetate into it, and hope that they start to produce methane. And then you transfer dilutions from that into these tubes. These tubes are also anaerobic sealed. They are oxygen free, and there's agar inside. And when you are lucky and you found a substrate where only the acetate oxidizing bacteria can grow on, then you get these small, tiny colonies. But this is a lot of luck, to be honest. And then you might be lucky to have a pure culture of these acetate oxidizing bacteria. And then you have also to have a pure culture of a methanogen, which you have isolated before. And then you put them together in a new bottle under anaerobic condition and look if they start to produce methane. And then you have proven that this syntrophy exists. We have only 2 examples for acetate oxidation, for example, worldwide. More we don't have isolated from these because it's actually more or less impossible. So this syntrophic interaction takes time. I want to show you that here. They have a long lag phase. As you see here, here is displayed the grow. This is a mesophilic culture growing at 37 degrees. And they start growing after 30 days. And this is good already. And then the grow proceeds, but they never reach very high level as they have little energy they can grow from. At the same time, they start to grow. You see that the acetate goes down and you see that the methane starts to be produced. And this lag phase you have here in the beginning, I want to call it establishing the thermodynamic window. And this is what you see here. No, this is another culture, sorry. This is an example from a thermophilic condition. Here it goes a little bit faster. They start growing around after 10 days, but you have also more energy in the system at 55 degrees, slightly more at higher temperatures. And as you can see here in the pictures, they're forming like flocks when they grow, as they need to grow together quite close to each other. And they're forming these flocks to enable a good transfer of the hydrogen between the bacteria and the bacteria and the archaea. So what does that mean? This thermodynamic window they have to find. So you see here an acetate oxidizer, here's the methane producing bacteria, and here's the hydrogen. This one is producing the hydrogen from acetate, and this one picks up the hydrogen and produces methane. And this bug likes hydrogen a lot because it's growing on that. It wants hydrogen, but this one needs to get rid of the hydrogen because otherwise acetate oxidation doesn't work at all. So they have to find a level where they have low enough that this can grow, but still enough that this one can grow. And here's a more scientific expression of that. You can see when the hydrogen pressure becomes less and less and less and less, this acetate oxidation turns into a negative value, means it starts to release energy. But you have the opposite for the methane producers. They work with very high hydrogen levels, very good, and they can grow up to a certain level hydrogen, but when you go even more down, they can't grow anymore. So the thermodynamic window is very, very small. So now it's something, how, from what do they live? How do they conserve actually energy? You can see that this reaction, the acetate oxidation and methane production, it only releases that little energy. But what you need to produce an ATP from that, what is the currency of energy in a cell, you need much more, almost double that much. So it actually means that there is not a single reaction in the whole pathway which would gain any ATP. This is not possible at all because the low free energy does not allow ATP production directly. So what else do they? Here I put a very simplified pathway from the acetate oxidizing bacteria. The acetate is converted to CO2 and to hydrogen. And the hydrogen is picked up by the methanogens together with CO2, which produces methane. And what they have in these systems is this ferredoxine and this NAD. And they have to get rid of the electrons that put it on protons that produces hydrogen. Here it's the same. They put the electrons on the protons producing hydrogen. And when you look in this table, this is actually what happens in syntrophy. This is the free energy you get from oxidizing ferredoxine, and this from NADH. So this is plus, close to zero. This is even more worse, has even a higher plus. But when the partial pressure of hydrogen goes down, if you reduce more and more the hydrogen, what is done by the presence of the methanogens, then it becomes negative, and then there's energy suddenly in the system. So how do they gain energy then from that if they cannot produce any ATP? So I brought you a picture here where you can see two enzyme complexes they have in the cell. These are so-called hydrogenases, one sitting in the membrane, and there are also one system which is in the cytoplasma. And what happens here? They take this ferredoxine and this NADH, transfer the electron of proton, and they use the free energy to pump protons over the membrane. Or they consume protons on the inner side of the membrane. Both together creates a proton gradient over the membrane. You might have that in your lectures. It creates altogether a so-called proton motor force. But what it actually means is that you pump slowly out proton by proton. Whenever you have energy, one proton is pumped. That means it's the smallest fraction of energy actually, which you need to produce this gradient over the membrane. Proton for proton, they pump out. And when they have enough collected outside and the gradient is big enough, they open the gates here. This is the ATP synthase. And they let flush it back. And this is like a waterfall. There is a lot of energy in, and this energy can be used to produce ATP. And I think this was my last slide. Yes. So I hope that you get some information how differently biogas can be produced, that renewable energy is not using renewable energy is not always means not always sustainability, that the microbial process behind is quite complex and complicated, and that it lives from a process which is called syntrophy, and the syntrophy is very special and a lifestyle which let the microorganism live on a very close limit on, or very close to the limit of thermodynamics, and that they have extremely problems to gain energy, but they are specialized on that and make a good living on that. So I thank you for your attention. And I hope you didn't get headache from the last 3 slides here.
Very cool. I've always been amazed by the thermodynamics and how they can actually, with such low amount of energy, they can still find a way
Yeah.
to eke out a living.
And this is the way they found actually. This is collecting these small fractions of energy by just pumping proton by proton. If they have enough, they can gain an ATP from it. So you only need that much energy to make one pump. And this is valid for all the syntrophs actually. They have all these systems. Very, very, very fascinating. And also these are fascinating hydrogenases. They can use both actually at the same time, which makes it possible that you can use the NADH actually as well. Somebody else, a question?
Microbial life is complicated.
It is quite complex, and it's a lot of interactivity between the microorganisms. And I mean, we have studied them by isolating them and characterizing them, but meanwhile, of course, we recognize that we cannot just look at them separately. We have to look at them all together to understand what they really do.
Exactly. We touched upon that last week, discussing Yeah, and risk assessment and bioremediation, it's far more appropriate to look at microorganisms as a community and how they're working as a community, because you always have them working in one function, which is then coupled to another function.
Yeah. And they're also, I mean, they depend on in many other ways on each other. So they pick up things other produce for them because they can't produce themselves. So they actually connect to each other by that, getting amino acids provided they might not be able to synthesize. And they share, actually. They share to, at the end, perform something together.
I don't know if you saw that, the last thing I'll say. It's funny you mentioned the swamp gas there, the will-o'-the-wisp, because I did a little demonstration at the beginning of the course where I went out to, it's called Dalkarlskäret. It's a swamp that's a bit further outside of Uppsala. And I was able to collect enough methane where you could really just watch it flame right up.
Great, great, yeah.
It was cold, but you know, it was
Yeah, yeah.
always interesting.
You did this, or?
Yeah, I
Yeah.
basically I did that.
Yeah, yeah.
Exactly.
You see a very old experiment, very simple, and you still can use it to demonstrate.
Mhm.
I need to say I never saw a flame myself burning, but I am not so often in the dark in the swamp, so probably it's the reason.
You don't spend your time in dark swamps.
Mm.
Why not?
Yeah, maybe all the ecologists are doing, making studies, I guess.
Yeah, yeah, we spend a lot of our time in dark swamps. Yeah, exactly.
I have a question.
Yes.
In the swamps, is it dangerous for people to be there for a long time? Because it's, I mean, for the oxygen?
No, not really with respect to the methane. The content is actually low, but you also have, of course, hydrogen sulfate, which can develop. And this is much more dangerous for the human health. But you can smell it. And this is the positive thing. The methane you can't smell.
No.
But the hydrogen sulfate, you know that you are close to something, you should go away.
So it usually is hydrogen sulfate and methane at the same time?
Yeah, it often occurs together, yeah.
Okay.
But it's mainly more methane at that moment, but still very small and tiny amounts of hydrogen sulfate smells already.
Okay.
But where you have a lot of methane, methane development, you have less hydrogen sulfate, but you still have it, but in small concentrations. But already the small concentrations are smelling and are already dangerous and can harm the body.
Okay, thank you.
There was that also in the same clip. I found a spot that did have very high production of hydrogen sulfide, and I could really only stand there for a very short period of time because it smelled so bad.
Yeah.
And it was, you knew it was starting to get, you started feeling a little funny, so I had to leave. But that there was so much organic matter in that one region because it had this big vegetative mat that was floating over a good portion of water, and you could feel it bouncing up and down as you were walking on it. So there was a very large hydrogen sulfide production. That's usually from the breakdown of the proteins, isn't it?
Yeah, it is from the protein. Mainly, yeah. I mean, as to the moment you have, it depends how much proteins you have, but then you have also, when you have sulfate reducers, you also get this. And I mean, the sulfate reducers, they usually can use the hydrogen CO2 better than the methanogens, but still they exist next to each other. But this is with the ratio of the concentration, what I mean. Even small amounts of hydrogen sulfate smells a lot. So if you have just very little there. In the biogas system, you have hydrogen sulfate, what comes from the proteins and not from the sulfate.
Yep. Yeah, it's sort of the essence of sulfur cycling.
So I hope you got something that was not too complicated at the end, but syntrophy is difficult to explain and to understand what it actually does. and how much they depend on each other.
Okay. Well, if there's no other questions, I just have a couple of announcements. So if you don't mind, Bettina, I'm gonna
Yeah, do proceed.
move screens. This won't take very long. So I just wanted to first remind everyone about the quizzes. And then remember that we have an exam coming up, the 12th, 14th of March. There'll be instructions posted on Canvas, but just a heads up, it's going to be 7 questions, short answer. They're going to be based on the lectures, but you should also have a good handle on the literature, because they're going to be, you know, sort of these short answer questions that are kind of jumping over a bit on the, so it makes you sort of think and have to look up the answer a bit, so to synthesize the information. So just keep that in mind. There will be, like I said, I will post all the instructions on Canvas this week, but the questions won't open up until the 12th. So you'll have the weekend to do the questions. It will be time limited. The group reports, that should be sent in the 12th of March. I know maybe there's not so much happening with your columns. How is there anything happening with your columns? Are they doing something?
Nope.
Nope.
Mine is farting if I press it.
It's what? Sorry?
It's farting.
Yeah. Okay.
Well, then something is going on. So it's producing quite a bit of gas then, and I assume it smells kind of bad. Yes, no, it's farting. Okay, so there's that hydrogen sulfide. So, okay, well, then there's something going on. I guess maybe not with the sun being so dim still, there's not much color change though. Well, there's at least some activity then between the farting and, I mean, like I said, this one, it's changed slightly. There's a nice big red ring at the bottom now. So as of now, the group reports, they should be sent in on the 12th. We've had a few people that are sort of leaving the course, so I might rearrange a couple things with the groups, just so you know. That will be done like by tomorrow. So there might be, I need to double check with a few people here just to make sure, so people aren't kind of left by themselves and not have to worry about taking on too much of the burden for the group reports. But remember to go through the instructions. So there's that. And then the final thing for the course will be the course days. And I put up the schedule, but maybe just to remind everyone, the ideas that I had for the first day was a few presentations from you guys, one being your egetarbetet, so your own work. And really that's going to be fairly short. Just keep in mind to prepare like one PowerPoint slide going over the interesting aspects of your organism that you chose for your work. And then also a presentation of the columns. So it'll be interesting to see pictures or even see them via Zoom. And you can have maybe 2 or 3 PowerPoint slides for that. And the home lab supplies will be sent out the week before, so you'll have all that equipment at the ready, plus the instructions, which should be up by the end of the week. So you'll have that. And I believe that is pretty much all I had to announce, but are there any questions of any kind? Does it seem straightforward? Okay, like I said, I'll get all the group stuff sorted out by tomorrow for the group reports. And then I think we are set for tonight. Next week, we have our last lecture. Well, last lecture for the module. And that will be presented by Sarah Halim, and she will talk about microbes in water purification. So how we keep our water clean. And that falls over many different aspects, industry, wastewater treatment. So there'll be a few different things to think about in that one. Okay, then I thank you for this evening. And Bettina, everyone give a round of applause for Bettina. Thank you very much for this. It was a very nice lecture.
I was happy to tell you something about biogas processes.
We really appreciated your time tonight.
You're welcome.
Okay, guys, then you'll be hearing from me via email a bit more and Canvas. And if you don't see me beforehand, you will see me next week. All right. And the literature for next week will be up tomorrow.